Welcome to this talk, and I'm so pleased to welcome back Dr. Clare Craig. Clare, thank you so much for coming back.
Thanks for having me, John.
Now, we're sharing some amazing animations today from I haven't actually learned to pronounce his name properly, I'm afraid, Erik Tangsoo in Estonia, filmmaker. And he's kindly given us permission to share some clips, which we're going to play now. But just before we do that, if you want to read the best book that's ever been written on the COVID pandemic as a summary, in my view, it's this one. It's still available, Claire. You can buy this on Amazon.
Absolutely, yeah.
Yeah. So do get that. We've done a couple of videos on this in the past and covered it in great detail. Anyway, without further ado, let's go on to the first clip. And this is really quite amazing. It teaches about science, pharmacology, cytology, and it just brings everything to life, basically. So let's give it a go. So we're looking at this first clip now from Erik. So we see a needle releasing lipid nanoparticles. And then we see all these lipid nanoparticles. So that's presumably sticking in your muscle somewhere and squirting in lots and lots of lipid nanoparticles. So just before we look at the circulation there, Claire, these lipid nanoparticles, I did a quick calculation once, and it seems that each vaccine dose contains thousands of billions of them, probably several trillion of these lipid nanoparticles. So they're absolutely tiny things.
They are, and I think that there isn't something we can compare them to particularly. It's a new technology, and the tininess of them it needs to be understood in terms of other things in the body, because you can't because they're much smaller than anything else that we have sort of dealt with in medicine. And so obviously that gives them different properties. And one thing I would say is what they do when they're making it is they put the mRNA into the solution, and because of it being charged, the mRNA gets sort of encapsulated by the differently charged lipids. So it sort of forms itself in the solution. But if you leave it over time, these tiny little lipid nanoparticles, they can coalesce with each other and become bigger. So over time you might get a whole variety of different sizes and therefore doses in any one lipid nanoparticle. But so this video is sort of the pharma story of what they ought to have looked like.
Yeah, these are the ideal lipid nanoparticles, and I've actually got a diagram I've just produced here. It's pretty basic, but it shows a red blood cell. So the big red circle is the red blood cell, and the small dots are the lipid nanoparticles. And the AI tells me that this line across the middle, that you can get 87 lipid nanoparticles will fit across one red blood cell. So we're assuming that the red blood cell is about 7 micrometers, that's 7/1000ths of a millimeter. And these lipid nanoparticles are probably around about 80, 90 nanometers. That's the next one down, Claire, isn't it?
Yeah, thousandths of a micrometer.
Yeah, thousandths of a micrometer, yeah. So do you know Erik, Claire?
Yes, I do know Erik. He is a very talented man who's worked incredibly hard. He's made several videos, like documentary videos about vaccine injury, and he's recently produced these graphics with a friend, as I understand it. But yeah, he's very motivated, and it's really interesting seeing how being motivated by truth can get these things out in a way that, he's not being paid to do this. No. He's really not got money as a motivation at all.
Yeah, it's proper intrinsic motivation, isn't it? He's done it because he believes it's the right thing to do, which is brilliant. And he's produced 2 films, actually. One's called Inside the mRNA Vaccines, and another one's called Inside the Vaccine Trials, both of which, of course, we'll link below in the description. And these graphics we're looking at today come from Inside the mRNA Vaccines, but this is just, generously has given us permission to share these just for general teaching purposes, which, of course, is remarkably generous. So this is the bit we've seen, Claire. So we've injected these lipid nanoparticles. Is there any physiological equivalence of sort of lipid nanoparticles in the body? I mean, when we were given these lipid nanoparticles, will the body sort of say, "Oh, these are lipid nanoparticles, I know what they are," or is it something new?
It's new, and I was just thinking through when you said that about what we don't know, because the problem is, we've got electron microscopy that can show things that are really very small, but I'm not sure it can really resolve down to a few nanometers like that. So I'm not sure we've ever been able to know what's there at that scale, actually.
Yeah.
It sort of becomes, you end up working on crystallography and things like that, which we know, which we have observed less with. And yeah, so of course, the thing is that the body is set up to deal with things that are foreign, and we have all of these things that have evolved to protect us, but they require the foreign thing to behave in a way that foreign things normally behave, right? So that they would come into the cell, by binding onto a receptor and hijacking the cell's equipment. And when they get into the cell, there would be sort of replication and double-stranded RNA and things that our cells can detect as viral invasion. Whereas the lipid nanoparticles just, they get taken up in what's called endocytosis. So the membrane kind of takes a drink of the liquid that's around the cell and takes it into the cell. So it's an active process to bring it in. It's not just slipping in. But when it's in there, it's acting on the cell, and the cell doesn't necessarily have the mechanism to say, "I'm being hijacked," and alert the immune system.
Right, I hadn't fully appreciated that, Claire. So I'd assumed that the two cell membranes kind of come together, and the lipid part of the lipid nanoparticle stayed outside, but the whole lipid nanoparticle goes into the cell. So you end up with this foreign lipid as well as the mRNA actually inside the cytosol of the cell.
That's right, and I'd also made that mistake. I'd also assumed it was just sort of membrane fusion that was happening at the cell surface, and it does happen like in the endosome once it's in. But I think it's quite an important distinction, because a lot of people have got worried about cells that have had lipid nanoparticles in. If they're coming in just by sort of passing through a membrane, they could just as easily come back out again. And so there was this kind of fear of what's going in the other direction. But I think because it's an active endocytosis that's bringing it in, it's mostly a one-way street.
So do we actually know what happens to this foreign lipid material once it's inside the cell?
Well, I mean, actually, I just realised I was saying all this and sounding very definite about it. Of course, all of this is sort of based on, it is based on some work, but it's. I think you always have to sort of cage it with uncertainty. We don't know everything, do we?
No. I mean, we do have this process called phagocytosis, so we know that this process occurs.
Yes.
Materials taken inside a cell, but that we normally associate that with neutrophils and macrophages and defense cells, don't we?
Yeah. So the phagocytosis is where you're gobbling up material debris around the cell and then using the inside of the cell to destroy it. So you're sort of mopping up dead things or mopping up things that shouldn't be there. Whereas this endocytosis, all the cells do it, and it's a way for them to kind of they pick up sort of what's going on in the environment around them. It allows them to uptake other things as well that they can use to sort of see what's going on.
But of course, the macrophages and the neutrophils, that they have the destructive digestive equipment in their cytoplasm, just ready to get rid of all this debris, just get ready to lyse these bacteria and viruses. But ordinary cells presumably don't have as much specialised digestive destructive equipment within them. So these lipid nanoparticles...
They do have some though, they do have some, but they don't. But then, I guess that takes us on to the next bit. Well, maybe we'll come to that when we're looking at the mRNA, but there's the question of, once you've got something foreign inside a cell, what's it going to do with it?
Absolutely, yeah. And the answer to a lot of these questions is I don't think we really fully understand that, do we? I certainly don't.
No.
Let's look at some more video. So we're at a stage now where we've injected probably between 3 to 6 trillion of these lipid nanoparticles, we believe. Although we're open to correction on that number, but it's one heck of a lot, that's for sure.
It is. When you sort of put that into context about how many cells there are in the body, you're in the same ballpark now.
Yeah, that's interesting. I think the last estimate I heard was probably about 70 trillion cells in the body.
Yeah. So you tenfold out, only 1 for every 10 cells. That's quite an astonishing number. Yeah.
So we've got potential here. Given that we've got systemic distribution of lipid nanoparticles, and that's no longer a debate, and we'll see that in a minute, for transfection in multiple cells.
Yeah, I mean, I think we have to be realistic about gradients and concentrations and stuff, and there are some cells that are going to get hit a lot harder than others, particularly the endothelial cells that line the blood vessels. They get exposed to a lot. As well as, obviously, the idea was it was going to go into the muscle in the arm, right? That was the idea. Those are the cells that were meant to be targeted, which is an odd thing in some ways if you just sort of take a step back and pretend that you knew nothing about, like, vaccine ideas. So why would you want to tell the body to attack muscle cells? Like, is there a respiratory epithelium you're meant to be trying to protect? Anyway, Moderna did a study, I think it was in 2023. They got funding to do a study to say they were going to look at what happened to the lipid nanoparticles that did not go into muscle cells. And in that publication, they stated that only 2% did.
Wow.
So they sort of were saying, it's very inefficient trying to get it into muscle cells, so we have to give you an awfully high dose. You're like, well, about the other 98%. So they say, well, we're going to find out what it does. You're like, really? In 2023?
Incredible. Let's look at some more video. Phenomenal graphics.
Yeah. I mean, this kind of graphic work takes a lot of effort.
Oh, heck yeah.
Yeah, they've done a beautiful job.
Yeah, and we see them circulating.
And you've got all those blood lining cells that are going to be exposed. And of course, the exposure is not going to be uniform even within a blood vessel, because if you get any kind of microclotting anywhere, or rather when you get into the very, very small vessels, everything's flowing much more slowly there. So that's much more of a chance for it to get to the edges of the flow of the blood and interact with the cell. And if you've got any microclotting, then you're going to get stagnation around that clot and more of an opportunity to enter a cell as well.
It didn't occur to me before that if you get like an aggregation of lipid nanoparticles, you're going to end up with a bit of a blob, and that in itself could be that could constitute a blockage in the small vessels, and that could potentially aggregate, presumably, clotting factors to it.
Yeah, so if they get really big, and they do get really, really big. So I don't know if you've seen the videos where people have taken mRNA vaccine liquid, put it onto a glass slide and looked at it under a microscope. And when you do that, you start to see coalescing to start with. Like, obviously, it's happening at a scale your eyes can't see, but it gets to a scale where it becomes microscopic and you can see it coalescing. And then they start to form rectangular crystals, which are cholesterol crystals, because most of the lipid in a lipid nanoparticle is cholesterol. And when cholesterol crystallizes, it looks really inorganic. You get right angles, perfect right angles, with the rectangle usually with the corner missing. Or you get, if you look at those in the other dimension, they look like needles because you're cutting them across the other way. So the point is that if you heat it on the light from a microscope for long enough, you start to see the whole thing coalesce into crystals, which are relatively enormous. And so it all depends, I guess, on how long they've been there, what temperature they've been at, as to how much coalescence has gone on. And if you get the big ones, then probably less likely to end up in a vessel unless it's injected directly into a vessel, which can happen. So Mark Girardi's done a lot of work on this. Do you know his bolus?
Mark Girardi, yes. I've talked to him several times,
Yeah. So the whole idea being that you've got that sort of when you inject into a muscle, you're putting in a liquid into a space where there wasn't a space. So you have a high pressure, like balloon of liquid, and when the needle goes in, you will break small vessels with the needle. So depending how near that needle is to the high pressure balloon of liquid, you're going to get a different dose entering the vessels, and it can be that the whole thing just gets squeezed right in, or it can be that it's diffused over a slower time. And I guess the size of the lipid nanoparticles might alter what gets into the vessel as well. I don't know what scale I'm on when I'm saying that. I might be wrong.
But people as well, many videos I've seen of people giving these injections, they seem to take a pride in how quickly they can do it and how painless it is. And of course, the more quickly you squirt it in, the higher the pressure is going to be and the more small blood vessels you're going to disrupt.
That's right. And I mean, it's the thing about Mark's work, really, the thing that sort of really hits home with me is when he's looking at studies from a long time ago, because this is something that's really, there was a huge amount of research on. And so that kind of idea around pulling back on the syringe before you plunge it all in, and whereabouts in the arm you should be aiming to increase the level of safety, was because they knew it was a problem for decades. In the 1950s, it was well established that 2% of people will have a deltoid injection hit a vessel with negative consequences. So, if you're going to accept 2% negative consequences, that you need to have a very, very large benefit to outweigh that risk.
Don't tell anyone this, Claire, but in 1975, my charge nurse told me how to find out the gluteus maximus, how to stick the needle in, and how to draw back. And this was ingrained into us like drill. Stick the needle in, stabilise it, draw back, and only then inject if you're not getting any blood. And yet, for some reason, these guidelines were changed. It's just inexplicable to me that all of the vaccine injections that are now being given are being given wrong because people aren't aspirating.
I mean, but realistically, the people who were injecting in those vaccine centres weren't they weren't qualified nurses.
Indeed.
They were volunteers.
But in the last few years, I've worked with qualified nurses who've told me that you don't need to aspirate when you're giving vaccines. I mean, I did, but the qualified staff are actually being taught the wrong thing by the national guidelines. It's rather bizarre.
Yeah, that is bizarre.
Yeah, it is. So basically what we're talking about so far, we haven't even started talking about the mRNA yet. All of these complications would occur purely because of giving these foreign lipid nanoparticles, even if they contained absolutely nothing inside them.
Well, that's right. And so lipid nanoparticles were worked up originally for gene therapy, right? So there's this idea that, if you've got a child usually, but, some of them are adults who've got a missing gene and they need that gene, and they need it in a lot of cells around the body. So they say, well, how can we get material to as many cells in the body as we can? And so that was the kind of impetus behind lipid nanoparticle technology. And when when BioNTech was sort of established, this was their realm, was, delivering genetic loads into cells. So they were looking at the lipid nanoparticle technology, and they were complaining about the toxicity of it because they knew you couldn't give repeated doses, because whenever they tried, there was too much toxicity associated with it. So they said, well, it's no good really for gene therapy because of the toxicity, but we could use it for vaccines because then you only need one dose. And of course, look at what's happened. People are 9, 10, I don't know what they're up to now, but they knew it was toxic.
Yep. Let's look at some more video. Yeah. Of course, the lipid nanoparticle, you see the lipid nanoparticle there, but of course we know the scale is tremendously smaller than the red blood cells we see in the background. I think this is just showing the idea that we've got systemic circulation of the lipid nanoparticles, Claire, with the blood.
Yeah, I quite like that as well for showing the flow.
Yeah.
That, if you've got a vessel that's got fast flow, then the chance of your lipid nanoparticle hitting the edge and interacting with the cell is lower. So, it's when you've got a stagnation somewhere or a smaller vessel that it's at most at risk.
And of course, in some capillaries, thinking particularly about the brain and the lungs, the capillaries are so small that the red cells have to squeeze down, don't they, to get through them?
Yeah, I've seen that under a microscope where they're all deformed and trying to get around the corners.
Yeah. Now, this next clip, Claire, I think we're showing this very process you've just educated me about, the endocytosis. Endo in cell, cytosis, cell going into the cell. So we see a whole lipid nanoparticle being subsumed into a living human cell, albeit in animation. So we've seen the lipid nanoparticle go in, and we've seen it deliver its payload of RNA.
Yeah, which is of course not where RNA should be in a cell. RNA in a cell should it's in the nucleus. It gets made in the nucleus, and it comes out into a part of the cell called the Golgi apparatus, where the proteins are made.
Yeah.
And so it's got a position to be in the cell, but this is coming in from the membrane and is sat in the cytoplasm.
Yeah, it'll go to the ribosomes, then onto the Golgi to be exported, won't it?
Yeah. And I don't know how much we know about what happens to random foreign mRNA. I mean, obviously we know it makes protein, so some of it's getting into that Golgi apparatus and is being used. But you think, well, how did it get there? Because the cell, it doesn't travel on its own. It's got to be moved, and so the cell has all this equipment that moves things around.
Yeah.
Something's moving it to there. And I don't know. Some of it potentially is getting destroyed, or it's getting spat out. But some of it does end up making protein.
It's presumably translocated in the endoplasm via the endoplasmic reticulum, one would assume, this network inside cells.
But how does it get to the endoplasmic reticulum?
Indeed. Indeed. Yeah.
So I think a lot of the kind of concerns that we're expressing here about what happens in a cell have just been sort of brushed aside on the basis that that cell will be destroyed. So if the cell is going to express a foreign protein, it will be destroyed. And that's true. It's true. And that's problematic, when it's just gone into that endothelial cell. If you destroy endothelial cells because they're expressing spike protein, then you create all the things you need for an environment where a clot will form. And if it's a tiny one endothelial cell, that will recover. But if you have a patch of endothelial cells, the recovery, like a skin wound, comes from the outside in. And so if it might take longer, and then you might get a more serious clot forming. But the thing is that there are circumstances, I think, where some of those cells haven't been destroyed. And that's why we've ended up with people who are still producing, where you can still find vaccine mRNA in their bodies now.
Yeah. Something just occurred to me. If, and we know it's not true, but if it were the case that the mRNA vaccine just remained in the deltoid, we know it's not the case. But if it were, then the RNA would go into the deltoid muscle cells. The deltoid muscle cells would express that foreign protein. Those deltoid muscle cells would then be attacked by the body's own immune system. That's going to make your arm weaker, isn't it?
Yeah. So
if you're killing muscle cells.
Yeah, I think Mark did a kind of sum with me on this when we were saying, if all of those 6 trillion went into one cell each in your arm, how much arm do you lose?
Pretty well all of it.
You lose a litre of arm, we figured out in the end.
Yeah.
I mean, obviously that's not what happens. And any one cell will have multiple going because, they're grouped together. But we were just sort of playing with the maths of
Playing with the numbers. I must say I'm not as good at press-ups as I was, but there could be other factors involved in that. Yeah, so here we see a lipid nanoparticle going along a small vessel, and then it just goes into the endothelium of the blood vessel. And it's just disappearing inside. So when we're thinking about the development of blood clots, Claire, is the condition and quality of the endothelium, the lining of the blood vessels, an important factor in keeping blood liquid or in facilitating the start of a blood clot?
Both. So the endothelial cell produces like little anticoagulants that just say, keep everything flowing and smooth and can't make it so it can't clot, but also in order to have a clot, you need to have some sort of nidus for that clotting to begin on. So the absence of an endothelial cell creates that. It's a wound that needs a clot. So you've got both. You've got you lose your anticoagulation and you produce a wound.
So it's a double whammy. You lose the anticoagulant effect of the healthy endothelium, and then that damaged endothelium itself becomes potentially thrombogenic.
That's it.
Yeah. This is Vircho's, isn't it? Vircho's triad, from what I remember.
Yeah, Vircho's triad has got the. I think you're going to test me now. Has got
it's the vascular endothelium, the viscosity of the blood itself, and the turbulence of the blood flow.
The flow, yeah, yeah.
Yeah, neither of which we want, of course. Immune evasion. So this lipid nanoparticle here is containing the mRNA, and that means, to my mind, that the body's immune system can't get to this mRNA. Because we've got foreign mRNA, normally the body would destroy that.
Yeah, well, so the foreign mRNA and DNA naked is highly immunogenic, like dangerously immunogenic. It would cause an anaphylactic response in people that don't normally have.
Really?
So yeah. So that's one of, obviously they are using the lipid nanoparticle to deliver it into a cell, but also to protect it from the immune system.
Yeah. So really, we're conning the body's immune system here to ignore something which is normally very, very immunogenic.
Yeah, we are.
It's all just hidden.
Yeah. Whereas if it was a virus particle, when that's in the blood, it looks foreign to the immune system.
Yes.
Whereas a lipid nanoparticle has no protein on the surface that looks foreign. It's just lipids. So it can get it bypasses all the sort of protective things that we have. And of course, being an injection, it's already bypassed multiple layers.
Yes.
We're breathing in things all the time, and we have all sorts of layers of protection for what we breathe in.
Yep.
But if you inject, then it's in regardless.
Yep. And also, normally, if there's superficial bacteria, that they'll be taken up by the lymphatic system and go to the lymph nodes. But if you're injecting, you're bypassing the body's natural lymphatic defences as well.
Well, yeah, I mean, the point about the deltoid injection was to, the sales pitch was that it would get into those phagocytosing cells that are picking up debris that live in the muscle.
Yeah. And
they would go via the lymphatics to the lymph node and educate the immune system in the lymph node. So that was the sales pitch, was not that the public were told that, mind you. That's what they were telling the doctors, was it was going to get to the lymph nodes. The public were told it would stay in the arm. Yep.
And this also is very important for the DNA contamination, because we now know that the vaccines contain DNA contamination. And I believe you're going to correct me on an error I made shortly on this, but we know that there's DNA contamination. And the levels for DNA contamination that are allowed inside vaccines were actually created, that those levels were created assuming that it was a naked DNA that was being injected.
That's right, yeah.
Which should be quickly got rid of.
As you said before.
Yeah. But now we've got the DNA, foreign DNA, that is protected by these lipid nanoparticles, so it can go straight to our body cells where it can potentially do damage.
Yeah, and one thing we haven't mentioned is that when they were sort of trying to get permission to do this, they had done animal work showing where in the body these lipid nanoparticles went. And as part of that work, there's a submission to the Australian regulator, which included images where they had done immunofluorescence of individual cells to show where it was going within a cell, as well as which organs it reached. And you can see it in the Golgi apparatus. It's lit up green on the immunofluorescence next to the nucleus, but you can also see some in the nucleus. So the lipid nanoparticles were entering the nucleus, and that again, they are designed to. That's kind of what the sort of primary design of lipid nanoparticles was. Now, to be fair, that primary design has evolved into many different types of lipid nanoparticles. So you can kind of pick your tailor-made one that has propensity to reach different parts of the body and different parts of a cell. And these ones are not ones that are designed to go into the nucleus. But that image showed that nevertheless, the nucleus wasn't entirely immune from them.
So the lipid nanoparticle itself, or components from the lipid nanoparticle, were getting into the very heart of the cell, into the nucleus of the cell.
Yeah, and again, actually, that doesn't matter if the cell gets destroyed.
Right.
Doesn't matter, does it? It's dead.
Right.
It matters only if the cell is somehow evading destruction. And that, I think there are parts of the body where these lipid nanoparticles can get to more easily than immune cells do. So in the testes, in the brain, you've got these protected areas where we don't let the immune system come and attack, because we don't want to. The brain's got to be protected, and testis is only the cells in there are producing half of your DNA, so they have a sort of more foreign look to them. You don't want them attacked, but the lipid nanoparticles could get in there.
Yeah, but they wouldn't be attacked by the full force of the immune system, because those areas have to be protected. Yeah, makes perfect sense.
Yeah, I mean, we say that, but of course, in some people they were. There is a definite signal in the vaccine adverse reaction databases for testicular inflammation, swelling and pain. So some men did have highly symptomatic testicular inflammation after their injections.
Right, and that I haven't read that in the normal lists of side effects, but that's it's concerning. And Dr. Ros Jones, of course, has been doing some work on fertility effects. But if there was an injection where you've got naked DNA, then that's much more likely to give you a severe allergic reaction, Claire, is it?
Oh yeah, it is, and that's one of the causes potentially for the anaphylaxis.
Right.
And then also the technology involved large vats of E. coli being used to produce the DNA template, and endotoxin, which is this bacterial cell wall from the E. coli.
Yeah.
Is something that's really quite dangerous to inject, even at teeny tiny levels. It's almost not a dose response. Even the smallest level of endotoxin can cause problems, and the problems that most people are familiar with are dramatic. Anaphylaxis is dramatic. You don't miss that. It's happened, but there's very good evidence that it causes many more inflammatory problems, including chronic problems, which are harder to measure and easier to miss and easier to not associate because of the timing.
Yeah. And of course, it's endotoxins that give rise to a lot of the features of sepsis, life-threatening condition. Yes, endotoxins are, our bodies react very badly to endotoxins. They are quite dangerous at scale. And we're not talking about a sort of sepsis level of exposure here. This is a very small amount. But even that small amount can be problematic. Yeah, given the choice, I won't have any.
No, and the thing is that there are, that kind of manufacturing of a vat of E. coli at scale has many kind of economic advantages. And it means that you can do things like, what they do is they put an antibiotic resistant gene into the bacteria that they're using, and they can use that antibiotic and kill off anything else and just select out the ones they want. So that technology is great for them. And it used to be used to make insulin. That was how it was originally made. Yes. And so diabetics were at risk of having that endotoxin problem. And that's all been moved over to using yeast instead, where there are other issues, but there's not that endotoxin problem. And using yeast is slightly more expensive, and it has other advantages and disadvantages. But I just do find that interesting that that it was rejected for insulin production.
Very interesting. Yeah. Now I think what we're seeing here is a an illuminated lipid nanoparticle that's been labelled so we can see it. Yeah, luciferase.
Okay,
Yeah.
luciferase. Yeah, I was going to say they've chosen the right colour, because that is, luciferase is the gene that was used so that the lipid nanoparticles fluoresce under that immunofluorescent microscope. So when you see the nucleus, when I was saying you can see it got there, it's that colour on the when you get the images.
And of course, this is a bit of gene technology, because this luciferase is actually present in certain things that glow in the dark, isn't it? Like deep sea fish. And various insects that I can't remember the name of. But
it is, of course, named after Lucifer, the light carrier.
Yes. We'll pass on from that one. You might.
Yeah, I think we should.
There might be some negative connotations in that. Yes.
I mean, it does get used a lot in science as a marker because of that ability to visualise it afterwards.
I mean, it is a useful scientific tool, isn't it? It lights things up and you can follow it. So it's very good.
Right.
Yeah. So we can follow those lit up nanoparticles as they are systemically distributed around all of the body. And when we say all of the body, we mean all 5 toes, your ears, and every single organ in every part of the body. It goes absolutely everywhere. And potentially with the lipid nanoparticles there as well. So this next video, Claire, we're looking at the modified RNA here. So this is not a natural physiological ribonucleic acid, is it? It's been jiggled around with.
That's right. So natural, our own mRNA, which is messenger RNA, that's what the M stands for, because it takes a message from the DNA to make it into protein. And it lasts half an hour, maybe an hour or two, and then it's gone. So it's I liken it to being I say if you imagine you're going to do some cooking in the kitchen, so you're making your proteins, and that you go to the computer database to get a recipe. So that's your DNA. And then you print it off on the printer. And that's your RNA. And then, by the time you've finished cooking, or maybe I'm a messy cook, but the paper's kind of rubbish and you throw it away. So that's like an analogy that I like to use to describe how it normally works. But the M here is for modified RNA. So, or at least it can be for modified. It's sort of, being used interchangeably in a slightly kind of dubious way. So there was one company called CureVac who used sort of pretty regular RNA with the same lipid nanoparticles that Pfizer and Moderna were using. They did not get the same results. And I mean, there's various reasons why that might be, which are not all to do with the product they were using. But it was all attributed to that. So what Pfizer and Moderna did is they took out one of the nucleotides. So you've got the 4 nucleotides that make up the letters that make up the sequence. They took one of those out and substituted it with this 1-methyl-pseudouridine nucleotide.
And pseudo, of course, means false. It's not the real uracil. It's not the real uridine. It's a false one.
And it's something that we don't have. We do have a tiny, tiny amount of it normally. But it's not something we have very much of at all. And here suddenly, all of the every 4th letter almost is this strange chemical. And what that strange chemical does is it stops the RNA being destroyed. So it sustains it for a longer time in the cell. So it sort of acts like a longer dosage. So you get more protein produced from it. And that there was a time when I was concerned that after this mRNA was broken back down into nucleotides, that there would be a situation where we had in our cells too much of this abnormal one, and it would be getting used in other protein manufacturing, and that those mRNAs would hang around for too long, and the proteins produced would be too much, and it could cause all sorts of problems. But I was wrong on that. So the pseudouridine, the body doesn't use it, just rejects it.
That's reassuring. But it's good that you erred on the side of caution there, unlike other groups that could be mentioned who perhaps have not erred on the side of caution.
I mean, I didn't publish what I thought about that. I just was thinking about it.
No, but it makes perfect sense that you could end up with this naked pseudo-uridine that could be a building block for future RNA molecules potentially, but thankfully that hasn't transpired to be a real problem. So here we see the RNA code, and in the background all these letters are the A, U, C and G. The adenine, it should be uracil, but it's pseudouridine, cytosine and guanine. And here we see all these letters lit up that are a synthetic base in the RNA. David Speaker actually described this as making it plasticky, that it hangs around for a long time.
I think it's quite a good description, yeah.
Now, we've got these pseudo-uridines here in the sequence. And basically, any 3 of these bases in this sequence will code for one protein. It's called,
one amino acid.
Yeah, sorry, one amino acid, yeah. One amino acid, which is a component of a protein. So 3 code for an amino acid. Now, the idea is that if there's a pseudo-uridine, the base reading apparatus in the ribosome might not recognize this and might skip one. Is there any basis to this frame shifting idea, do you think, Claire?
Oh yes, absolutely. But all of that evidence came out after everyone had been injected with it. So although actually that's not entirely true. Now I think there was knowledge about it beforehand, and the evidence in this specific scenario about what exactly the frame shifting might look like was published after the event. And the publication showed that 30% of people who had been injected had antibodies to one particular protein that you might see if the mechanism slipped when it was reading the letters, which is what the frame shifting means. So it's like your hands have moved on the keyboard and you're typing a totally different word.
Yeah.
And but the thing is that was just one of the proteins. There's any multitude of different things that could be produced, and or different lengths, or different, all sorts of things can go wrong if you've got machinery that's just, not aiming properly on a keyboard, which is what was happening.
So if you frame shifted, the 3 bases would read for the wrong amino acid. And you'd be putting the wrong amino acid in, therefore you'd end up with the wrong protein, and you could potentially end up with pathological protein production.
That's it. So you're just producing gobbledygook. And we know it didn't happen entirely because, people did create antibodies to spike. So they were, there was a kind of, there was the majority, or at least a significant amount, was doing what was intended, albeit whatever the intent is, whether it was making the spike protein, but there was other proteins being produced. There's undoubtedly other proteins being produced, and shortened spike proteins where the RNA had got fragmented, so you didn't have the whole story. It just cuts short.
Components of a spike protein rather than the whole thing. Yeah.
Right, and the thing is with proteins, their function is all about the shape that they're in. So the sequence of amino acids isn't really the thing. It's how they fold into each other to produce a shape that has a function. Yeah. And if you have a shortened one, that means that the interactions of them with each other when they're folding will be different, and you could produce something that's a very, very different shape and a very, very different function, even though it was nearly, an entire spike protein. So there was all sorts of things being made for which we can't be certain what the function is. We don't even know what the sequence was, and consequently, a lot of attention has been put on the pathology being related to spike, whereas in reality the pathology was so diverse. Yes. I think partly because it wasn't just spike.
Well, if 30% of people were developing antibodies to a foreign unknown protein, mean, those antibodies would only be developed by the immune system if that protein was present. So the antibodies are actually a marker for the presence of this abnormal protein.
Yeah, which we still know nothing about.
Yeah. So I think what we're seeing here is this is the RNA, isn't it? Being read by the ribosome, and the ribosome is producing the this sort of wedge-shaped spike protein.
I like that graphic. I think you can see how that could stick onto the shell of a virus and look like a spike.
Yeah.
And there they go. And I think actually that image is quite useful as well, because although, like, in theory they wanted the spike to be in a cell that would get destroyed and therefore educate the immune system about destroying cells with spike.
Yeah.
But some of the spike got out of the cells and was circulating in the blood. And that was particularly bad with the AstraZeneca, but it did happen, I think, with the others too.
I mean, that's what you'd expect with production of foreign proteins, really, wouldn't you? You'd expect some to stick to the surface of the cell, but you would expect some to become humoral and to be distributed in the body tissues.
Yeah, and I guess that brings us back to this thing I was saying earlier about how things move in a cell. So there are these things called chaperone proteins whose job it is to transport so when a protein comes out of the ribosome, it gets taken to where it's needed. It gets taken to the cell surface, or taken to the nucleus, or taken to wherever it's needed for its job.
Yeah.
And these, I mean, maybe somebody does know, but I don't know what was going on there once this spike was produced. So you've produced a whole load of a foreign protein inside the cell, and some of it got to the membrane and stimulated an immune response. Some of it maybe got destroyed in the cell and then shown on the membrane to say, I've got this inside me, and am I for destruction or not? But I imagine some of it could just have been, well, if you've got something foreign, let's just spit it out, because that shouldn't be here. So the cells can, as well as sort of endocytosing, they can exocytose just whatever's in the cytoplasm that shouldn't be there.
Yeah. Yeah, and again, exocytosis is a well-recognised sort of cytological process. This is nothing new. So we've got here, Erik is saying here about a 10% error rate. And I guess what we're seeing here is a pathological protein that's being produced as a result of this error.
Yeah, so I would put it at a high percentage. I think once you've included all the mechanisms for error, so the fragmentation of the RNA, the fact that the RNA sequence could be read backwards as well as forwards, which is really abnormal. Normally,
I never thought of that.
Normally if you try and read a sequence backwards, you hit stop codons again and again and again. So that, sorry, I've just jumped ahead, haven't I? So each of the three letters can translate into an amino acid, or it can translate into a thing called a stop codon, which stops the process. And there's quite a lot of three-letter combinations that are stop codons. So normally when you turn a sequence back to front, you hit stop codons and nothing really happens. But with this particular sequence, there was this really odd absence of stop codons. So you've already got the kind of, well, you could do it the wrong way around error rate, and the fragmentation error rate, and then you've got the slippage error rates, and there's another one that I can't quite remember.
I had no idea these things could be read backwards. That's another sort of fairly concerning area.
Yeah, I mean, they shouldn't be, because each end of the sequence has got the sort of apparatus to say I'm the front and I'm the back.
Yeah.
But if it's fragmented, you'll be missing those ends.
Yeah. I mean, it's supposed to be, I think, just over 4,000 nucleotides in the messenger RNA that's given, but if a bit's chopped off, it would change the message.
Yeah.
Oh, here we see the activity in the nucleus, I guess, Claire. The reading of this is the nucleus. We've got the reading of DNA here. We can tell it's DNA because it's double-stranded.
Yeah.
So I guess this is the DNA reading enzyme or protein called polymerase II.
I think that's right. It's making the RNA.
Yeah.
So I think the point with this DNA contamination is, well, what was the DNA sequence of, and does that get made into RNA once that's in the cell?
Yes.
So, and obviously, if it can be delivered into the nucleus, there's a risk that it ends up in that cell's DNA, and that does happen. It's not like that's just not complete scaremongering.
No.
Our cells do end up with bits of foreign DNA in them, and then our cells die, and it doesn't matter. But over history, our DNA does have evidence of bits of virus being incorporated into it.
Oh, lots of it. Some people say sort of 8-9% of the human genome was originally viral.
And that's normally viruses that most of that is viruses that are DNA viruses, and that have the apparatus to get themselves into cellular DNA in order to be replicated. But it's not all DNA viruses, and there are ways in which RNA viruses can end up as DNA in DNA. And just, we've talked a lot about the mRNA vaccines here. I think it's worth touching also on the DNA ones. So AstraZeneca and Johnson & Johnson used a viral delivery system to get DNA into the cell. And so they were targeting the nucleus. The virus itself was targeting particular organs. It wasn't every organ, but it was quite a range of organs. The gut and the liver and... can't remember all of them. And the material went into the nucleus, but it was in the nucleus without being in the chromosomes. Like, that's what that virus does. It creates a sort of piggyback little mini nucleus within a nucleus. So in theory, that wasn't going directly into people's DNA. It was just being used within their cell nuclei. And again, if the cell's destroyed, it doesn't really matter either way. It's only when the cell isn't destroyed that you might get a problem.
Yeah. So if there's DNA contamination, whether it's from one of these adenovirus vector vaccines, or whether it's some DNA contamination in an mRNA vaccine, that DNA can potentially get into the chromosomal DNA, potentially. Does it need reverse transcriptase to do that, or can it just sort of diffuse in? I've never quite understood that.
Yeah, it needs to be made into DNA in order to do that.
So it needs reverse transcriptase.
Yeah, but the thing is that there are those types of enzymes around on occasion. It's not something we're completely immune from happening.
No.
It can happen at a low level, and I think people have shown it has happened now. They've shown in cancer cases.
Yeah.
That it was in the DNA in some cases.
Yeah.
I don't think people should panic that that's happened to them. I think that's always this interesting kind of psychology around all of this, is that people don't like to hear about harm because they took it.
Yeah.
And I just like to kind of emphasize that the harm is in a minority of people. And you, by sticking your fingers in your ears to what the harm might be, you're sort of prioritising your ego over helping address the harm that a minority of people have been affected by.
I agree, but I'm pretty angry that I've been conned into a game of Russian roulette that I didn't understand. So this picture here, Claire, this could be a physiological situation, isn't it? Could be with the enzyme reading the DNA, making the RNA.
Yeah, that looks healthy.
Yeah. Yeah.
But we don't know what that DNA sequence was. I guess that's the point.
Yeah, I think so, yeah.
The other thing, of course, is that we don't know what was happening with the bacteria in the gut. So there's quite good evidence that there was a problem with virus in the gut, in an infection.
Yeah.
And that some people end up with virus in the gut longer term in long COVID cases. So the bacteria seem to play a role in all of this, and I think that might prove to be the case in vaccine exposure as well, because bacteria love taking up DNA and seeing what they can do with it. It's like, they take up plasmids, it's their thing. And if it kills them, it kills them, but they're just constantly reproducing, so you've always got evolutionary drive. So I do think the gut may play a role, and that might also explain what I've kept referring back to about it shouldn't matter if the cells are dead, because if it's in the bacteria, then your immune system is living alongside those gut bacteria. It's not going to be attacking them in the same way.
Yeah. So here we see the spike protein on the surface of the viral particles. So this is a spike protein on the surface of the SARS coronavirus 2.
So the way that ends up there is that the virus has got a lot of instructions. I mean, not a huge amount, but more than just spike. And those instructions hijack other parts of the cell machinery, so that when it's replicated its whole RNA sequence, make a new virus, that gets carried to the surface to the membrane of the cell.
No, but this is the actual viruses we're seeing.
I know, but the membrane has already got spike on it.
Oh yes.
So as it gets extruded from the cell, the spike on the cell membrane encapsulates it, and it's off.
Oh, I
so
see.
like part of the viral replication works by getting the spike onto the cell membrane. That's how the virus is going to use it. So I don't know what that means in terms of when it's not got all of those different instructions around, but the idea that your spike would be on the cell surface might be integral to what kind of a protein the spike is, how the cell deals with that kind of a protein, is to stick it on a cell surface. Yeah, and of course it did circulate in the blood, but our immune systems are brilliant at dealing with foreign material.
Yeah.
So it was quickly removed from the blood. It wasn't like it's circulating for a long time. But in the immediate aftermath of an injection, you've got some in the blood, and then your antibodies would just be hoovering it all up.
Yep.
I mean, one of the things that bothers me about, I was saying about how the immune flags were bypassed, the things that say to the immune system, I'm being hijacked by a virus, weren't there. And one of the parts of the spike causes lymphocytes to hold off their immune response. There's a thing called PDL1, which it allows the sort of cell to kind of escape immune responses. And that happened with the virus, absolutely happened with the virus. And that's the reason why in an infection, compared to other viruses, although actually other viruses use all sorts of techniques like this to hide as well, but like it was taking a bit longer to get the T cell response to trigger. So the T cells were starting to learn what they were aiming for, but they weren't being told to fire. And so that meant there was more infection of more cells at the point where the immune system says fire. And so in some people, that delay meant that there was a lot of cell death all at once, and then you get the sort of cytokine storm reaction, you get the sort of toxic drops because you're killing a lot of lung cells all at the same time. So it was all about that immune delay. But that same bit that sort of downregulates PDL1 is in the vaccine spike sequence as well. And so, you're meant to be telling the immune system, come get me. I'm a vaccine infected cell. I want you to learn how to kill me. Come and get me. But it was included that part that says you can ignore me, which is also the same sort of signalling that cancers use to evade the immune system. So in many ways, the way these cells were evading immunity was similar, because it was, it didn't look like it was infected by something foreign. It just had a weird protein that seemed to have come out of it from it inside in a way that the immune system wouldn't understand as being foreign necessarily. At the same time as saying, you can ignore me.
Yeah. And I particularly love this one, Claire, where we've got a cell where the RNA has got into, and this spike protein is just produced on the surface of the body's own cell. So we now have the body's own cell.
And of course, there are some cells you do not want to mark for destruction. You know, if it's a respiratory epithelial cell, those cells are designed to have a short life expectancy because they will be infected, our bodies will kill them, and they will be replaced. And you know, so there is a turnover. Same with our skin surface cells, they get shed. So they've got a role in being something that dies a lot. But there are other cells in our bodies, like our brains and our hearts, that cannot be replaced at all. And so you don't want to lose any one of those cells, because that's it. And in the brain, obviously, you know, we want our brains intact. Thanks very much.
Yep.
And in the heart, when you've lost a heart cell, it gets replaced by scar tissue. And the heart is a very delicate electrical organ, so you don't want scar tissue in it, which can cause short-circuiting.
Yeah, and potentially lead to abnormal heart rhythms, as well as loss of the muscle. So you have the mechanical deficit, which of course can lead to heart failure if it's bad enough.
Yes, and we know those things have both happened. On a population level, there was a rise in cardiac arrhythmias, particularly AF, but all sorts of arrhythmias.
Yeah.
There's a rise in sudden cardiac death. There was a rise in heart failures.
Yeah. So here we've got the body cells expressing foreign spike protein. So this could be the endothelium here, the lining of the blood vessel. And instead of being nice and smooth, it's now all rough because it's expressing spike protein. And those spike proteins are saying to the immune system, better kill me, I'm infected with viruses.
That's right. And that whole, you can see there, they've got an area that's so infected that you're going to have a wound when all those cells are killed off.
Yep.
Which would be a clotting risk.
Yep.
Yeah. And in the infection, when you have an infection, the virus is pretty confined to the respiratory epithelial cells. So there isn't evidence of whole virus in the blood. There's evidence of RNA in the blood, but that's debris from killing them off. You know, that doesn't show that you've got whole virus. No one's ever shown whole virus in the blood. There was evidence of circulating spike proteins in the blood, which again is a kind of debris, you know. So, you know, I do think there were systemic effects from circulating spike proteins, but they'd get shut down quite quickly by the immune system. And so you can compare how much circulating spike there is from a viral infection to the vaccine circulating spike. And it's, you know, it's quite a frightening sum. I can't remember the exact number, but it's comparable. You're not, you know, the idea of a vaccine in theory is it's something inert, it's something tiny, it's just a little instruction, it's a little education, but the volume here is out of proportion to that.
Yeah. Interesting. So normally when you have a respiratory virus, you don't have a viremia, you just have viral fragments, and that's what gives rise to the fever and all the other systemic response effects. Yeah,
so there's a nice quote that I wish I had at my fingertips from a paper that Anthony Fauci co-authored around how there was no viremia. The mRNA in the blood should not be considered to be a viremia. It is just debris.
Yeah. And what we're seeing here is these cells are actually being destroyed by the immune system, presumably mostly by cytotoxic T cells, I would have thought, Claire.
Yeah, so there's two types of T cells. There's the ones that destroy cells that are virally infected, and there's the ones that are sort of the generals who are making the whole immune system be coordinated and have the memory of what's gone on.
Yeah. Are the antibodies acting as what's called opsonins, Claire? Are they like labelling?
So they'll be labelling the free spike in the blood.
Yeah.
And then, yes, they when they're bound to spike on a cell, that is enough itself to generate a cascade of chemical reactions that lead to like the insertion of kind of channels in the cell membrane that cause the cell to just be drowned and destroyed. And then they're also saying to the other cells of the immune system, this is a bad one.
Yeah, they're called perforins, aren't they? They actually perforate the cell and result in the death of the cell. So what we're seeing here basically is lots of cells in that particular tissue there have been destroyed. So that actually constitutes a wound, and that's going to be a site for, not only is this endothelium not producing its anticoagulants, it's actually going to be a pathological site which can trigger the coagulation cascade. And I think as well as that, another thrombogenic mechanism is the spike protein itself has been found inside the blood clots.
That's right. They could show that if you took blood and added synthetic spike that had been made, using that vaccine sequence to it, that created this weird kind of clot. I'm not sure we should call them clots because they didn't have, platelets weren't needed for it to happen. Normally platelets are really important in clot formation. But this just required the fibrin, which forms the kind of structure of a clot, and the spike itself. And there's these beautiful images that have been done on electron microscopy of what a natural clot looks like and what these clots look like. And the fibrin looks like spaghetti. And then you've got this parmesan all over it, which is the spike proteins that are causing it to stick together.
All right, so the spike proteins causing the fibrinogen to come out of solution, essentially, directly.
Yeah, directly. And of course, the thing is that, as well as having very delicate mechanisms for clot formation, we also have delicate mechanisms for clot destruction, because removing clots is a key part of keeping everything functioning well. And this is a foreign thing. The spike fibrin bound inverted commas clot is not something that those systems could remove in the same way.
Yeah, we have these mechanisms. Tissue plasminogen activator was the drug that we used to activate the plasminogen in the tissues, which actually breaks down the clots. Yeah. And so really we've got three mechanisms of thrombosis here. We've got the reduced natural anticoagulants that aren't being produced by the damaged membrane because the membrane is partly dead. It's not producing what it normally produces. We've got the physical wound effect, which is going to attract, potentially stimulate platelet activation. And we've got the actual direct thrombogenic effect of the spike protein itself. So we've got three mechanisms of thrombosis there. Yeah.
And of course, with we said we've got a clotting risk, right?
Yeah.
So if we had this in theory and said, well, what would you expect if you then injected a population? I think you and I would say you would expect strokes, pulmonary embolisms, heart attacks, and
DVTs,
plus more, right? And the evidence is that there was a bit of a signal on those things. What's difficult is those things are very common. So you can increase how many there are in a year, but to show a statistically significant increase takes a lot of disease, unless you're looking in populations that don't normally have those problems. So if you can focus on a young population and say, well, they don't normally have, strokes. How many strokes in young people were there? Then you start to get a better understanding of the risk. But that doesn't mean the risk wasn't there for the older people. It's just that the signal statistically isn't there because there's more background. But I think what was happening was a lot of this clotting was happening in very, very small vessels because of that thing around stagnation of the flow to get into a large number of endothelial cells all together, all in one place. And so it's the smaller vessels damage that leads to damage in all of the organs, because that creates an area of damage within whichever organ that little vessel is in. And so then you get a situation where there are people who, prior to all the vaccines, let's say they had a problem in their kidneys, and this problem in their kidneys they were completely oblivious to, and they may have lived the rest of their life without ever knowing. But then they killed off a load of healthy kidney cells, and then that kind of exposed a problem that they might not otherwise have had. So there's that problem of sort of cellular reserve, that you don't want to go destroying healthy cells, because they might be compensating for problems that you didn't know about. And so I think that is really important to understand, because if you understand that you've got problems in vessels, that could be in every organ, and that if you've got this problem with cell reserve, that you're killing off cells that are expressing spike, that can be in every organ. And so you start to see a pattern of lots and lots of different diseases apparently being caused by the vaccine. And our safety mechanisms are set up to say, we want to understand when there's a signal that's genuine and when there's a signal from increased reporting.
Yep.
So they say, well, if you've got things in all sorts of different organ systems, we'll ignore it, because that's just a reporting signal.
Yeah.
But in actual fact, the MHRA in the UK, they published that the sort of yellow card reports for all the other drugs. So they were claiming there was an increased reporting signal because they'd done such a good job at selling yellow card reporting around the vaccine, which is obviously something they ought to have been doing. But they didn't do a good job of it, because reporting for all the other drugs actually fell in that period. So, there was no increased reporting. It was the same doctors who were continuing to say there's a little problem with the drugs over here, and they were saying there's a big problem with this drug, but it's a problem that's affecting lots of organs. And when people managed to get hold of the data finally from America, they could analyse different the sort of reporting rate for different conditions. So I'm not saying there was no coincidental reporting. There's bound to have been some, but you can use the sort of level of coincidental, very low level reporting on some organs to say, well, how much more often were things like myocarditis being reported?
Yeah.
And you get that sort of ratio between the two. And when you do that, there were 770 different conditions that had a higher signal than myocarditis.
Wow. Incredible. And as well as that, the signal should have been lower, because a lot of old people had died, and the old people would normally suffer from more of these pathologies.
Oh no, but to be honest, I think if you're an old person and you've got a vaccine injury, trying to persuade a doctor of that is really, really hard.
Yeah,
really.
But I mean, a lot of old people died from things like midazolam and morphine, and
yeah, that too. Yeah. But I think what was happening with the reporting is people saying, oh well, of course there's lots of deaths and disease because we've injected lots of old people, so of course there are. But if you dig into the data, the reports are not disproportionately in old people at all. Yeah. Most of the reports are in middle-aged people who shouldn't be having these conditions, because then it's much more obvious to a doctor that something's wrong.
Yeah. And the other thing is that if you kill off a portion of cells in an organ, then you might not notice that when you're 25. Because it's like renal function. When you're 20, you might have, 2 times more renal function than you need. When you get to my age, you've probably got exactly the amount of renal function you need. But in 10 years' time, I might have a bit less renal function than I would like.
Yeah.
And if you've already killed off some cells, you lose that reserve. So we could actually, over the coming decades, get what are essentially accelerated aging effects.
Yeah, we might well do, because of course aging ultimately is just organ decrease, isn't it?
Yeah, and aging is just basically due to the fact that your stem cells have only got so many divisions, and once they're used up, that's it. And if you've destroyed it's a bit like Parkinson's disease, the idea that someone's exposed to an environmental toxin, it takes away their reserve. You don't notice it when you're young, but decades later, when you get normal cell sentience and death as well, you haven't got the reserve because they've already been bumped off when you were young. So you develop the symptomatic disease. It's
yeah, no, I am concerned about that. So I think to say something that's slightly a counter to what a lot of other people have said recently, in terms of excess mortality, I think we saw a wave of vaccine-related excess mortality, particularly in 21 and 22, with a bit in 23, and by the time you get to 2024, it's over, actually. And I say that because I managed to find an ONS population prediction for deaths. So they have to say, well, looking to the future for planning how many deaths are we going to see every year.
Yeah.
And this was done in 2018. So this population prediction for deaths, I think, is a fair way of saying how much aging is going to contribute to mortality over the time. And by 2024, we were back to roughly where we should be as a population as a whole. We weren't for younger people. There are still problems, but the bulk of that death wave as a population was over. So I think that we saw this kind of medium-term problem, but we're now in the long tail, and we don't know what that's going to include, because it becomes harder and harder to measure it and to attribute it to something that happened years before.
Correct.
But I think there will be a long tail because of the nature of these products. Now there's a clot.
Yeah, that's a blood clot.
You've done it with, I mean, in actual fact, there weren't many red blood cells in these weird clots. That's why there's that sort of white clot.
So the white fibrin platelet-rich clots. But they'll still block off the blood supply.
Yeah.
Yeah. So thanks very much to Archie for allowing us to use those quite incredible graphics. Yeah. Brilliant, Claire. Thank you.
That's right. Did you want to talk about that last image?
Oh, this one.
Yeah.
Yeah, go for it. Yeah.
So, well, John said he'd done this to get them to scale, so you could see that the virus, which is really one of the tiniest things, it's hard to kind of describe quite how small viruses are, but the lipid nanoparticles are another degree smaller than them.
Yeah. And can squeeze through little spaces that aren't supposed to be squeezed through. Yeah. So thank you so much for that, Claire. And I believe you're working on something quite interesting now. Are you working on something quite interesting now?
I am. I have been very, very occupied and busy over the last few weeks working on the autism story, which is something that, I got captivated by, frankly. It's sort of drawn me in, and there's an awful lot about it that I think we can understand much, much better than we have done so far.
And I understand you've got a video and some publications coming up, hopefully in the next week or two.
That's the plan. So the plan is to have a publication that can get peer reviewed, and then a video to explain it to everybody, and then launch it.
Well, there's only you and me listening, Claire, so you can tell us what it's about now if you like. There might be a few eavesdroppers, but I doubt it.
What I will say is that Dr. Tim Kelly is the one that got me onto this. He had this brilliant idea, and we've been working on it together. And it ends up being a sort of framework for understanding all the risk factors and how they fit together. And it has really big implications, which is why we're doing it slowly and carefully, and not just doing it bit by bit as we go along. Excellent.
We'll be waiting. So when will we hopefully get a release on that?
I'm hoping in the next few days.
Wow, fantastic! Yeah, and if you would choose to come back on and talk about it, we will of course be delighted. Thank you for that.
Love to. Yeah. Thanks, Tom.
Look forward to that. It's always good to leave on a cliff edge.
Yeah.
Thank you.
Thanks, Tom.